Hello everyone, I am Marco Palombo from UCL and this talk is about our work on large-scale analysis of brain cell morphometry to inform microstructure modeling of gray matter. I have nothing to disclose. Diffusion MRI is a promising technique for characterizing brain gray matter microstructure in vivo. Biophysical modeling is often essential to quantify changes of potentially important cellular features such as soma size, projection size, length, and more. However, to design sensible biophysical models and appropriate diffusion MRI acquisitions, it is essential to know what are the typical values of these microscopic cellular features. Despite the growing amount of microscopy data on brain cell structures now available publicly, there is still a lack of studies reporting a comprehensive summary of the typical values of these cellular features. In fact, only one study analyzed the cell reconstructions from microscopy data, but it focused only on features describing neurite orientation dispersion in gray matter. Therefore, the aim of this work is to provide the currently unavailable typical values of a large set of microscopic cellular features of interest for gray matter modeling. We downloaded and analyzed the three-dimensional reconstructions of about 3,500 brain cells from neuromorpho.org, representative of 8 cell types from 3 species. We measured 13 morphological features using custom scripts in MATLAB as we have previously described. We first measured the general features such as the degree of branching of cellular projections, the number of primary projections radiating from the soma, and the extent of the cellular domain. Then, by separating soma and projections, we measured the soma features such as the effective radius and the amount of soma surface covered by projection interfaces, and features of each branch of each cellular projection, such as mean branch effective radius, coefficient of variation along the branch surface to volume ratio, as well as branch length and tortuosity, and mean branch curvature and microscopic orientation dispersion. Our results show that at the cell domain level, brain cells have on average 6 projections radiating from the soma, which branch with a branching order of 9 to cover a cellular domain of 330 microns in radius. The cell body is on average 7 microns in radius, with less than 20% of the surface open to connect to the projections, and a surface to volume ratio of 0.5 over micron. Finally, cellular projections have branches on average 50 microns long, 1.5 microns in radius, and surface to volume ratio of 8 over micron, with a coefficient of variation of 1 micron. Branch tortuosity is on average 1.3, microscopic orientation dispersion 0.3, and radius of curvature 60 microns. A comprehensive list of all the features measured can be found in this table, also reported in the abstract. We conclude by providing an example of how this study can inform biophysical modeling. Considering the following typical acquisitions and a couple of arguably reasonable assumptions, based on the results from this work, we can hypothesize that restriction from soma can be significant, as shown by the gray area lying within the green area. The green area defines the combinations of timings for the assumed experimental conditions, while the gray area defines where we expect the measured diffusion signal to have sensitivity to the soma size estimated in this work. This supports previous findings. In contrast, restriction from cellular domain is likely negligible, as shown by the gray and green areas that do not overlap in this case. Longer timings are required to gain sensitivity to this feature, but then exchange may become significant. Similarly, impact from projection curvedness is likely negligible, unless longer timings are used, according to a recent theoretical work, as well as the impact of branching in line with previous findings. Remarkably, the undulation index measured for brain cells in this work is like the undulation index measured for white matter by Brabeck et al. We can then extend their conclusions to also gray matter and hypothesize that undulations can bias cellular projection diameter estimation when there is sensitivity to this feature. To summarize, we have provided the previously unavailable reference values of brain cell morphological features. This information is essential to build more sensible biophysical models of diffusion MRI signal in gray matter and to optimize data acquisition. Our results support some of the existing modeling assumptions while challenging others. Thank you.